Hello, I am Sam Smith, and this is Sam's MCAT Basics. This podcast covers the most important topics put out on the AAMC MCAT each year, and I determined this list by going through all the official practice materials that the AAMC puts out, and also some third-party practice material, and just put together this big list of topics that consistently kept on showing up, and ranked those topics and made podcasts out of it. This podcast covers enzymes, and I'm going to cover kind of a wide variety of topics that deal with enzymes. The first is going to be enzyme kinetics, and then I'm going to talk a little bit about enzyme inhibition. I'm going to go over the basic types of enzymes and talk about classes of enzymes, and then I'm going to talk a little bit about restriction enzymes. What are they cut? What are they useful for? Etc. So. I hope you enjoy. I hope this is useful for studying. All right, so let's begin with a brief definition of the term enzyme. So an enzyme is a protein or sometimes RNA catalyst that accelerates biological reactions. So enzymes act on substrates, which you can think of as reactants, to create some product. And they make reactions faster by lowering the activation energy. And if you've seen a reaction coordinate or the diagram of as a reaction proceeds, you'll see that this activation energy you can think of as like a big hump, and you can see this hump kind of decreases after you add an enzyme to a reaction. And it's important to note that an enzyme increases both the forward and reverse reaction rates. And just like all other catalysts, enzymes do not affect the thermodynamics of a biological reaction. And by thermodynamics, I basically mean how much of a given product will form during some kind of reaction. So it has nothing to do with thermodynamics, everything to do with kinetics, or the speed of a reaction. I'm going to repeat this point because I think it's important. An enzyme increases the reaction rate. It increases the speed of the reaction, but does not do anything to the amount of product formed at equilibrium. And think back to general chemistry and remember that equilibrium just refers to the extent a reaction proceeds at time equal to infinity. So if you left a reaction sit alone forever, this is the amount of product that would be formed. All right, so now that you have the basic definition of an enzyme down, let's talk about a few different models of enzymes, or models of how enzymes function. So the first model I'm going to talk about is called the lock and key model. So in the lock and key model, the key refers to the substrate, which again is the reactant, and it goes on to say that the substrate has a specific shape or arrangement of functional groups or other atoms that allows it to fit like a lock and key into the enzyme. The second enzyme model is called the induced fit model, and so this says that partial binding of the substrate causes conformational changes in the active site, and this allows the enzyme to fully bind to the substrate. And it's important to note that there is no correct model. Enzymes can use either of these models in order to function, and I'm sure there's some enzymes that use even more complicated models, more complicated mechanisms than what I've been describing. For the purpose of the MCAT, just know that both of these models are plausible. There's no one is correct, the other isn't. Enzymes can function either way. And so the structure of a substrate pocket determines its function. And so if you go back, listen to the amino acid podcast, I get into detail on this, in a lot greater detail on this. But basically, these functional groups that are on amino acids stick out into the substrate pocket and either form the shape of the substrate pocket or go on to form different functions that this enzyme needs in order to convert the substrate all the way to the product. And it's also important to note that enzymes, when heated or when introduced into some pH or pH that's away from what the normal pH range they function in, they can denature, and this leads to dysfunction. because it disrupts internal bonds or internal interactions between amino acids. All right, so let's get into enzyme kinetics. So the Michaelis-Menten model or Michaelis-Menten equation, which I'm sure you've heard of in probably biochemistry, is the main model used in calculations involving enzyme kinetics. It relies on a few basic assumptions, of which I'll get into a little bit later in the podcast. So the Michaelis-Menten equation goes like this. V naught, which is the initial velocity of an enzymatic reaction at some substrate concentration, is equal to the maximum velocity times substrate concentration divided by Km plus the substrate concentration. And so 2 quick important points before I go over each of these terms in this function is, number 1, this function is hyperbolic, and number 2, V naught is a function of substrate concentration. So V naught is the initial reaction velocity. That is the speed of the reaction, usually given in moles per second or millimoles per second, that occurs initially, right when the reaction starts. It's boom, right away. It's not talking about, 10 seconds later. It's this first initial portion. And this is something to remember because, you kind of think of the Michaelis-Menten equation and you think, okay, you see these curves and you think that this is the speed of the reaction, as you add more substrate. But it's really just referring to the speed of the initial reaction, not necessarily, it's not going to be the same speed the whole time. It's going to slow down as you use up substrate. But just remember, it is initial velocity. And then, so moving on, we have Vmax, which is the maximum initial velocity. So you can think about it like this. At a certain point, the addition of substrate does not further increase the initial reaction speed, right? This is an asymptote on that the plot that you see, and that is known as its maximum velocity. In other words, any additional addition of substrate does not increase your initial reaction velocity beyond Vmax. Then you have your concentration of substrate, which pretty easy, usually in units of micromolar, that I've seen at least. And then you have the Michaelis-Menten constant, and this is Km. And so this is the amount of substrate needed for an enzyme to obtain half of its maximum initial reaction velocity. So this is definitely the most important variable to understand. Again, it is the substrate concentration in which you have reached your half Vmax. So let's talk real fast about the information that your Km tells you about your enzyme. So the most important thing to know that your Km tells you is the binding affinity. So a high Km corresponds to a low binding affinity. And you can think about it this way. So when you have a high Km, that means that the amount of substrate that you need to add to a reaction in order for that reaction to be at its half maximal velocity is really high. So you got to dump in a ton of substrate in order to get to your half maximal reaction rate. And so, on the other hand, when you have a low Km, this corresponds to a high binding affinity. And so that is, you only need a little teeny amount of substrate in order to reach your half maximal velocity. Your enzyme and your substrate have very high affinity for each other, and therefore, you don't have to throw in a bunch of substrate because your enzyme just eats that shit up. It just boom, 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 boom. So that's how I remember that. And it's important to note that your Km is not to be confused with Ka or Kd. And these are binding association constants or binding dissociation constants. And you're definitely going to see this on the MCAT. So listen up. So the Ka is just an equilibrium expression for your enzyme substrate complex. So your Ka, again, association constant, is equal to the concentration of your enzyme substrate complex over the concentration of your substrate alone and your enzyme alone. And then your dissociation constant is just the reverse. So that's the concentration of your enzyme alone times your concentration of substrate alone over your concentration of enzyme substrate complex. And so each of these kind of corresponds to what you think it would. So, your association constant, if you had a high binding association, that's just high affinity. That means you're going to have a ton more of the enzyme substrate complex, higher binding affinity. And on the other hand, if you have a let's say a very high dissociation constant, that means they're going to want to be disassociated, low affinity, they're not going to be bound together. So again, let me just recap that real fast. So high Ka, high binding affinity. Low Ka, low binding affinity. High Kd, dissociation, likes to dissociate, that's a low binding affinity. However, you have a low Kd, that's going to be a high binding affinity. Your enzyme substrate complex forms at a much higher rate, or equilibrium favors the formation of your enzyme substrate complex, i.e. high binding affinity. Another very important term to know is KCAT, which is, as it sounds, a big K with CAT as in cat as the subscript, and this is also called the turnover number, and this is the number of substrate molecules each enzyme converts to product per unit time. And so it's also equal to Vmax over your concentration of enzyme. So remember that, Vmax over concentration of enzyme. And KCAT can be used to calculate catalytic efficiency, which is something you also may see on the MCAT. And so catalytic efficiency is your KCAT over Km. And so I remember catalytic efficiency by thinking about how you would expect these things to affect catalytic efficiency. So what do I mean when I say how I would expect KCAT and Km to affect catalytic efficiency? Well, when you have a high KCAT, you are probably more likely to have a higher catalytic efficiency. Why is this? So if you remember, KCAT is Vmax over concentration of enzymes. So the higher that is, that means that for lower concentrations of enzyme, you're going to have a higher Vmax. In other words, the higher that KCAT is, the better your enzyme is going to behave, right? Because that's just saying that for my teeny amount of enzyme, I'm getting an even higher Vmax, and this is a great performing enzyme. So I remember that is on top, because if the better the enzyme is, the higher the KCAT is, and so therefore higher KCAT will be higher catalytic efficiency. Km, on the other hand, has an inverse relation to catalytic efficiency. Lower Kms mean better enzymes, because for lower amounts of substrate, you're going to be at that half maximal velocity. It's already going to be performing well at these low substrate concentrations. So that is how I remember that Km goes on the bottom of that expression and Kcat goes on the top of that expression. So again, catalytic efficiency, Kcat over Km. So before I go further, I want to talk about conditions in which Michaelis-Menten applies, or I want to go over these assumptions that are made on which Michaelis-Menten relies. So there's 4 conditions in which Michaelis-Menten applies. The first is that only initial rates of the reaction are measured, i.e. there's no product that's been built up. And I kind of mentioned this earlier when I said that the function itself is V naught, which is the initial reaction. And then number 2, the second assumption is that the enzyme concentration is much, much lower than the substrate concentration. And the third assumption is the first steady state assumption, and it assumes that the formation of enzyme substrate complex reaches equilibrium quickly, basically saying that a steady state enzyme concentration is reached rapidly. So you hit that, you hit a pretty much stable concentration of enzyme substrate complex very quickly. Then we have the fourth assumption, which is also called the second steady state assumption. And
Before we get into it, I just need to preface with a little bit of information. So you can imagine the enzyme and the substrate binding, and then some reaction occurs within that enzyme and the enzyme substrate pocket to convert the substrate to the product. And then the enzyme and substrate basically break apart or disassociate, and now you have your enzyme, which can be reused, and you have your product. Now, the fourth assumption, which is the second steady state assumption, says that the breakdown of your enzyme substrate complex into enzyme plus product is assumed to be slower than number one, the formation of the enzyme substrate complex, and number two, the breakdown of the enzyme substrate complex to form your enzyme and your substrate again. So, it could happen that your enzyme substrate meet up, they bind, but then, the reaction doesn't happen to convert that substrate into the product, so then you just disassociates back out and you have enzyme substrate. So again, the fourth assumption says that the breakdown of substrate of the enzyme substrate complex to form enzyme plus product is slower than the formation of number one, the enzyme substrate complex, and the breakdown of the enzyme substrate complex to form enzyme and substrate. And just keep in mind that these assumptions aren't going to be heavily hammered on the MCAT like you will be after you take it. But I think it's important to have a little bit of an understanding of the Michaelis-Menten, obviously for the MCAT. I wouldn't just focus super crazy hard on the assumptions, but just know that this equation relies on a few major assumptions.
And so moving on, I want to talk about the different kinds of plots that are related to enzyme kinetics. So you're bound to see some kind of plot, and this might be a normal Michaelis-Menten, and by that I mean a hyperbolic shaped plot, or it might be linearized in a form known as the Lineweaver-Burke plot. And so, like I said, the Lineweaver-Burke plot is the linearized form of the Michaelis-Menten equation. So some people might memorize the form of the Lineweaver-Burke equation. However, I just like to remember that to convert the Michaelis-Menten, so all I do is memorize Michaelis-Menten equation. To convert that equation to the Lineweaver-Burke equation, you just take the reciprocal of both sides. So you take 1 over the left side of the equation and then 1 over the right side of the equation. And you get that into a form of y equals mx plus b, just basic middle school algebra. So you'll have 1 over v naught is equal to km over vmax times 1 over substrate concentration plus 1 over vmax. And now what I recommend you do is go back to the Michaelis-Menten equation, take the reciprocal of both sides, and kind of do this on paper, and you'll be able to see that you'll be able to actually derive that equation. And so when I'm taking the test, don't memorize that. I'll just whip out 1 over each side and then simplify it, and then that way I can take that and say, all right, here's the slope of the Lineweaver-Burke equation, which is equal to m, and then here is the y-intercept, which is your b term in the y equals mx plus b. And it's important to know the slope as well as the y-intercept, or what those correspond to, because a lot of times on the MCAT you're going to be given basically Lineweaver-Burke plots, and then say, here's enzyme X, and then it's going to show you a different line, which is enzyme X plus some inhibitor, and maybe the slope increases. Well, so then you look back at your equation, you say, okay, so slope is km over vmax, and that increased, so km increased. This looks like it's a competitive inhibitor or whatever. So very important to know the slope and the intercept. And of course, when you do that, take both sides, 1 over both sides of the Michaelis-Menten equation. Super easy. All right, so next I'm going to get into enzyme inhibition, and then I'm going to talk a little bit about how these affect the different variables of Michaelis-Menten, and talk a little bit about how that affects the graphs. All right, so there's two main types of enzyme inhibition. The first type is reversible inhibition, and the second is irreversible inhibition. And the reversible types are the ones you're really going to want to know, but I will cover a little bit of the irreversible. You may not see them on the MCAT. Not as important, but I'm still going to cover those. So reversible enzymes attach to enzymes in a non-covalent manner. This might be things like attaching to enzymes via hydrogen bonds or hydrophilic interactions or ionic bonds. And on the other hand, the irreversible inhibitors attach covalently to the enzyme, usually in the substrate pocket. And there are four main types of reversible inhibition. The first is competitive, and that is inhibition by competition at the active site, or the inhibitor binds at the active site. You have non-competitive inhibition, which is when a inhibitor binds away from the active site, and it doesn't. And in this case, for non-competitive inhibition, it doesn't matter if the substrate is bound yet. And you also have uncompetitive inhibition, and that is when an inhibitor binds to the enzyme substrate complex and locks it in. And so this does depend on the substrate binding first. When you're comparing non-competitive to uncompetitive, just remember competitive depends on the substrate binding first. And then last, you have mixed inhibition, and this is a mixture of competitive and uncompetitive inhibition. And so the inhibitor may bind to the enzyme whether or not the enzyme has already been bound, but it has a greater affinity for one state or the other. And so that quality right there is what differentiates it from non-competitive inhibition. The fact that it has greater affinity for either the enzyme substrate complex or the enzyme without the substrate bound. So that's the important difference to remember there. And so let's talk a little bit about the effects that these different types of inhibitors have on the Michaelis-Menten equation and the different variables in Michaelis-Menten. And you're definitely going to see a few of these on the MCAT. So first, for competitive, remember, this is an inhibitor that competes at the active site. And so for this, the Km, or the Michaelis-Menten constant, is increased. So you can think of that as lower binding affinity. So when you add an inhibitor, a competitive inhibitor, right, you're going to be competing at that site, and it's going to appear as though that your affinity for your actual substrate decreases because you have, this dude coming in, kicking out your actual substrate. So Km increases, and your Vmax remains unaffected. And this is because you can dump in enough of your actual substrate that you eventually get past that inhibitor, or like the inhibitor doesn't even affect it anymore because you have so much of your substrate. And you can outcompete the competitive inhibitor. Hope that makes sense. All right, and let's see. So the second type of inhibition is non-competitive inhibition. And so for this, the Km is unaffected. That's because your inhibitor binds away from the active site and therefore doesn't really mess with your binding affinity. And your Vmax is reduced because you can't outcompete it. And you once that inhibitor binds, you can't bind your substrate. Let's see here. So, and lastly, I'm going to talk about the uncompetitive inhibitor. So the uncompetitive inhibitor will actually reduce Km. And now, why is that? So if you remember, the uncompetitive inhibitor binds to the enzyme substrate complex and locks it in. And so a reduced Km corresponds to an increase in binding affinity. And that's because obviously when you're locking it in like that, it looks like, like shit, this thing is really just getting in there and binding, and it's not letting go because it has just such high affinity for that substrate pocket. But that's all an illusion, really. It's because of that inhibitor is just jamming it in there, binding it in there, and not letting go. So Km is reduced, and then Vmax is also reduced because obviously the reaction velocity is not going to go right if you're not releasing, if you're not completing that reaction and releasing the product. And quickly, let's talk about what this means for the linearized Michaelis-Menten, also called the Lineweaver-Burke plot. So for the competitive inhibitor, you're going to have an increase in slope once you add that inhibitor. And if you remember, the slope is Km over Vmax. And again, you add that reversible inhibitor, or you add that competitive inhibitor, and your Km increases. However, your Vmax is unaffected, and so your y-intercept is going to remain the same. So to sum that up, for competitive inhibitors, the slope of the Lineweaver-Burke plot increases, the y-intercept stays the same. And for the non-competitive inhibitor, which binds away from the active site, you have an increase in slope as well as an increase in y-intercept because Vmax is reduced and the y-intercept is 1 over Vmax, so it increases. And then you also have a Km that is unaffected. Yeah, Km is unaffected, and so your slope increases because the slope is Km over Vmax. And lastly, we have uncompetitive inhibition, and for this, the slope remains the same because your ratio of Km over Vmax is going to be the same no matter what. And then you obviously have a reduced Vmax, so your slope is going, or your y-intercept is going to increase. So again, for uncompetitive inhibition, your slope remains the same, but your y-intercept increases. All right, so let's briefly mention these irreversible inhibitors. And so irreversible inhibitors usually have some kind of reactive functional groups that are able to covalently bind to your active site. And this prevents, this basically deactivates your enzyme. That substrate pocket is no longer available for anything to get into. It's completely blocked. So there's 3 types of irreversible inhibition I want to talk about. So the first is called group-specific, or the first are called group-specific reagents, and these covalently link to specific side chain groups in the active site. So this is usually, hydroxide groups or amide groups or amine groups, sticking out from, for instance, serine or threonine or tyrosine, any of those amino acids that have these side chain groups that stick into the active site. So the second group of irreversible inhibitors are called affinity labels, and these are structurally similar to the substrate, and however they're not the substrate, and then they covalently attach to the active site. And the third and kind of most interesting type of irreversible inhibitor is called a suicide inhibitor. And so this is an inhibitor that is converted to an intermediate in the active site, and then that intermediate covalently binds to the active site. So, this molecule, I'm assuming, looks pretty similar to the actual substrate, and then something happens where you start to convert it to that product, and boom, it just binds right in there, and it stays in there. Yeah, kind of interesting. So last thing I want to talk about here is a term that you're going to see quite a bit, and that is allosteric inhibition. What is that? What is that? Sounds like kind of it sounds a fuzzy term. I'm sure you've heard it before. Maybe you do know what it is, but allosteric inhibition is the regulation of an enzyme by binding an effector molecule at a site other than the enzyme's active site. And so this effector molecule binds at what is called the allosteric site instead of the active site. And so non-competitive inhibition is a subtype of allosteric inhibition. So in other words, not all cases of allosteric are non-competitive in nature. So all non-competitive inhibition is allosteric, but not all cases of allosteric inhibition are non-competitive. Just remember that. And there's another type of allosteric regulation called homotropic regulation, and that is a type of allosteric regulation in which substrate molecules act to regulate the enzyme itself. And again, these substrate molecules bind away from the active site, which, obviously kind of counterintuitive, kind of strange, but interesting. Another term you might see in terms of inhibition is feedback inhibition. And so this is when the products created by an enzyme feedback and bind away from the active site. In the case of when it's allosteric regulation, these will bind away from the active site. I'm not sure. There might be some kind of feedback inhibition that is competitive in nature. However, so getting back to the point, feedback inhibition is when these products come back, they feed back into the enzyme and inhibit that enzyme. And this happens in a ton of different biological processes. So a good example of this comes in the case of purine production, or production of purine nucleotides. And so this process begins when PRPP, or 5-phosphoribosyl 1 pyrophosphate, is converted to phosphoribosyl amine. And so this pathway is inhibited by AMP, GMP, and IMP, which are all nucleotides. So if you remember, this is part of the pathway for nucleotide production. So, you can see that these products feed back and inhibit this enzyme. All right, now that I have talked about enzyme inhibition, I want to touch on the last topic, which is enzyme types. All right, so there are 6 subtypes of enzymes that you will need to know for the MCAT. The first are called oxioreductases, and these catalyze oxidation-reduction reactions. That's pretty easy to remember. Now, the second type of enzyme you need to know is called a transferase, and this also does something that it sounds like it would do. It catalyzes the transfer of functional groups between different molecules. And let's see, number 3, the third type of enzyme you need to know is called a hydrolase. And so a hydrolase cleaves bonds using water. And then the fourth type is called a lyase, and that catalyzes the breaking of various chemical bonds by means other than hydrolysis, or without water. So a lyase, remember that it just breaks it apart without water. And number 5 is called an isomerase, and so that catalyzes the transfer of functional groups within a molecule, so it makes isomers. And last, you have an enzyme called, this is number 6, by the way, this is the last one you need to know. This is called a ligase, and so that joins 2 molecules together to make an even bigger molecule. And all these enzymes are relatively important to know for the MCAT. You might get a question where it shows you some substrate and some product, and it says, what kind of enzyme
was able to, catalyze this reaction, or, and it's pretty easy though. I mean, you'll see one molecule and another molecule, two substrates, and then you make one product. Oh, what is that? It's probably a freaking ligase. Right, so that's easy. What is more important, in my opinion, are to know some common biological enzymes. And so I'm going to talk about dehydrogenase, synthetase, synthase, and phosphorylase, kinase, phosphatase, and I'm going to give a few examples of what these do. All right, so the first biological enzyme is called a dehydrogenase. And so a dehydrogenase removes a hydride, which is just an H group, hydrogen. It removes a hydride from a molecule and transfers it to an electron carrier. So, and these are usually NAD plus or FADH. So dehydrogenase takes the NAD or the FADH and makes that into NADH, which is an electron carrier. And if you're thinking, wow, that sounds like an oxioreductase, then you are very smart and you are also very correct. A dehydrogenase falls under the larger umbrella classification of oxioreductase. The next biological molecule is called, or biological enzyme is called a synthetase. Synthetase. It's got an E in the middle of it, which differentiates it from synthase, which is the next one. Anyways, a synthetase links two molecules together using the energy from ATP. And it is an enzyme that plays an essential role in the metabolism of nitrogen. And it does this by catalyzing the condensation of glutamate and ammonia to form glutamine. And again, it uses the energy of ATP. And so a synthase, on the other hand, links two molecules together without the use of ATP. And so a good example of this is actually ATP synthase. And so this is an enzyme that creates ATP. So again, you're not using ATP to create ATP. So that enzyme creates ATP, and this ATP is then used in a lot of different other biological processes. And so this ATP synthase essentially takes adenosine diphosphate, or ADP, and an inorganic phosphate, which is PI, and combines them to make ATP. And again, this is the transmembrane protein that is the last complex in the electron transport chain. Moving on, the next biological enzyme you're going to want to know is a phosphorylase. And so phosphorylases catalyze the transfer of a phosphate group to a molecule from an inorganic phosphate. So an example of this is glycogen phosphorylase. So glycogen phosphorylase catalyzes the rate-limiting step in glycogenolysis in animals. And so it does this in animals by releasing glucose 1-phosphate from the terminal alpha-1,4 glycosidic linkage. The next biological enzyme you're going to want to know is called a kinase, and this transfers a phosphate group to a substrate from ATP. So the most important difference between a kinase and a phosphorylase is that a phosphorylase transfers this phosphate group, transfers a phosphate group to a molecule from an inorganic phosphate. Kinase, on the other hand, transfers that phosphate group to a molecule from ATP. And so a good example of a kinase is protein kinase A, and so this phosphorylates a bunch of different proteins and basically makes these proteins active. So like enzymes, protein kinase A might phosphorylate an enzyme, and then boom, that enzyme is activated. All
All right, moving on, we have another very important biological enzyme called a phosphatase. And a phosphatase uses water to cleave a phosphate group off a substrate. And this goes the opposite direction of the kinase and the, what is that called? The phosphorylase. So a phosphatase uses water to cleave a phosphate group off a substrate. An example of this is protein phosphatases. And so these dephosphorylates amino acid residues and deactivates proteins. So these act basically directly against a protein kinase. So that's just kind of a brief summary of the biological enzymes you're going to need to know. And, those are important for a lot of these biochem questions. Off the top of my head, I think it's going to show some different pathways. You'll see I think there's a lot of kinases in particular, but those are all important to know. So the last thing that I want to talk about in this whole podcast is restriction enzymes. So restriction enzymes are DNA cutting enzymes, and each enzyme recognizes one or a few target sequences and cuts DNA at or near these sequences. And cutting typically results in single-stranded overhangs, which are called sticky, and or sticky ends. They kind of have like an L shape. They're called sticky because they can snap back together with their complementary base that they were cut with, or other strands that are complementary to their sequences. And most of these restriction enzymes cut at palindromic sequences, and so these are sequences that are read forwards and backwards at the same as the same thing. So essentially, if you take one strand, you flip it around, and then it should match that top strand. And so I'm sure you've seen the tattoos that are palindromic. For instance, one of my buddies has a palindromic tattoo on his forearm, and so it says his last name, and if you're looking straight at him and he has his forearm, in a normal human position, it reads his last name. And if he was to flip his forearm around and have his hand, pointing outside away from his body, it says the exact same thing. It looks the exact same way. So that's a palindrome. And go Google that if it still is not making sense to you. Restriction enzymes have a lot of uses in biochemistry. You can build plasmids. You can insert genes into these plasmids. And then these plasmids can be carried by bacteria. You can produce proteins, etc. And another interesting application of restriction enzymes are that they detect mutations. So let's just say that you have a genome and it contains a bunch of recognition sites for some particular restriction enzyme. Well, if some kind of mutation happens and now this recognition site is mutated, the DNA is not going to be cut at this site. And so what happens is you run one type of restriction enzyme, and let's say you get 4 different fragments of your DNA genome, and if you get a mutation, one of those sites is mutated. Now you don't cut the DNA at that point, and let's say now you have 2 fragments, and so that there was some kind of mutation in that DNA. And so usually you'll see this as bands on a gel. So this might be talked about in a different podcast, but when you run DNA on a gel, it'll separate it by size. So you have a mutation, you're gonna have 2 bigger pieces of DNA because you don't cut it instead of 4. It's pretty easy to see on a gel. All right, so that concludes the podcast number 6, which was about enzymes. Hopefully touched on some things that you have yet to study or was helpful in some way. Take a listen to the next podcast, an important one about groups and sociological groups. And this will be my first one that's not some kind of science topic, which makes me a little sad, but hopefully I'll be able to get through that, and hopefully it'll be able to help you. All right, so now for my MCAT advice of the day. Today, what I recommend is doing a massive amount of cars. Try to do it every single day. This is by far my lowest subject, and if I would have been able to get it even a few points higher, it would have definitely helped on my MCAT score. Just try to do it every day. Really put a lot of emphasis on studying it. Don't just think, hey, this is something that I'm not going to be able to do. I'm not going to be able to improve on, because you can improve on it. It's just something you got to practice. So do a lot of practice. Maybe I'd even recommend doing an hour or two a day, every day. It might take your MCAT score to that next level. Come on, Murph. I'm trying to podcast over here, dude.